Characterizations of bacterial culture. One of the key factors used in the identifications of bacteria is the cultural features, sometimes referred to as colonial morphology. This describes several aspects of bacterial growth on a medium, including the size, margin, shape, texture, form, and color. Our topic outline. First, characterizations of bacterial culture in agar plates. Second, characterizations of bacterial culture in agar slant. Third, characterizations of bacterial culture in nutrient broth. Fourth, characterizations of bacterial cultures in agar slants. Our objectives, first, is to enable the student to distinguish features of bacterial culture on agar plates, agar slant, and nutrient broth. Second, to enable the student to recognize the advantages and limitations of cultural characteristics in identifications of bacterial species. First topic is characterizations of bacterial culture in agar plates. Pigmentation of colony are yellow, orange, black, pink. Buff, brown, red, and purple. Surface characteristic are contoured. It is irregular, smooth undulating. Concentric, ridges radiating from the center like spokes in a wheel. Radiant, marked with ring having a common center. Wrinkled, rugose, mucoid. Smooth, glistening, dry. Shape of the colony, punctiform. It is under 1 mm in diameter. Rhizoid, it is irregular branch or like in a character. Filamentous, long irregular placed or interwoven threads. Shape of the colony, circular, spindle, irregular. Size of the colonies, pinpoint, 0.5 to 1 mm. Pinhead, 2 to 4 mm. Surface characteristic, entire. Smooth, does not have notches. Filamentous, long, irregular placed or interwoven threads. Undulate, wavy, shallow indentations. Curled, parallel chain in wavy strands. Lobate, with wavy and deeper indentations. Erose, irregular notch.
Elevation of colonies. Elevation describes the depth of colony developed by microbes. These are flat, raised, ambionate, convex, effuse, and surface. Effuse, subsurface. Odor absent or resembling. Streptococcus aeruginosus, sweet, cake-like, caramel or butterscotch odor. Pseudomonas aeruginosa, fruity grape-like scent or smells like taco chips or tortilla chips to some. Escherichia coli, floral, flowery scent. Moraxella cataralis, smell pungent and semen-like when growing on choke agar. Haemophilus influenzae, smells floral, flowery on choke agar. Proteus mirabilis, smells like rotten chicken soup or broth. Lastly, we have streptomyces, smells like wet earth, dirt after fresh rain. Characterization of bacterial culture in agar slant. Amount of growth, scant or slight, abundant, moderate, none. Color of colony: luteus yellow color, subtilus white color, and creamy white colony.
Consistency: butyrous, butter-like. Viscid, growth follows needle when withdrawn. Membranous, thin, coherent like a membrane, brittle, dry, friable. Optical characteristics: opaque, not permitting light to pass through, opalescent, opal in color, dull, not glossy, translucent, some light passes through but not enough to permit visibility of objects viewed through growth, iridescent, exhibiting changing rainbow colors in reflected light, fluorescent, having one color by reflected light and another by transmitted light, lastly, glistening or glossy. Form: filiform, unieffuse, separate or confluent colonies all throughout line of inoculation. Effuse, growth extend in form along the line of inoculation. Accenulate, tooth or pointed margins. Beaded, separate or confluent colonies at about the half end of inoculation. Rhizoid, irregular, branched or root-like in appearance. Arborescent, branched, tree-like growth. Odor. Here are some examples of bacterial colonies that have distinguished smells of their own. Medium change, non or color change 2. Simmons citrate agar slant. It is used to distinguish and identify Enterobacteriaceae based on how efficiently they use citrate, their only carbon source. Colonies that can use citrate as a carbon source cause a pH increase that turns the medium's appearance from green to blue. Salmonella coloraceus and Enterobacter aerogenes are distinguished by rapid growth and the medium turning blue.
Characterization of bacterial culture in nutrient broth. Nutrient broth is classified as basic nutritive medium because it supports the basic needs of ordinary bacteria. Broth culture gives the bacteria present easy access to the available nutrients compared to static bacterial colonies. Gentle agitation to keep the bacteria dispersed through the medium during incubation can aid this access further. Examination of broth culture must include the following: surface and subsurface growth, amount of growth, sediment, and pigmentation. Surface growth: non or ring, located at upper margin adherent to the glass. Pellicle, a growth in form of continuous on the surface. Flocculent, a small masses of various shapes floating in the culture fluid. Membranous, like a membrane, it is a thin covering growth. Subsurface growth: non or turbid, it is cloudy. Granular, it has small particles. Flocculent, a small masses of various shapes floating in the culture fluid. Flaky, it has large particles. Amount of growth: none or scanty, little quantity. Moderate, moderate quantity. Abundant, abundant quantity. Sediment: none or granular, it has small particles. Flocculent, small masses of growth of various shapes. Flaky, it has separate flakes. Viscid, sediment rises as a coherent swirl. Pigmentation: we have non-pigmented and pigmented. In pigmented, we have gram-negative anaerobic bacilli, gram-positive cocci, aerobic bacteria, and anaerobic bacteria.
Characterization of bacterial culture in agar slants presented by Krystal Ingal. Pure culture can be kept in agar slants for a long period of time. The tiny entry and the restricted air inside the tube, which together reduce water loss and exposure to outside air, particularly dust, as well as other contaminants, significantly reduce the chance of contamination or killing the culture related to the medium drying out. Examination of agar slant culture must include the following: amount of growth, form of growth, liquefaction. Amount of growth: none, scanty, moderate, and abundant. Form of growth: filiform, beaded, papillate, villous, and arborescent. These are the forms of growth. Liquefaction: none or crateriform, napiform, infundibulum.
saccate, and stratiform.